We're trying to do a single cell analysis under 10 minutes. So first, let's import the required packages. After importing the required packages, we need to load the data. Your data MTX file, gene names, cell barcodes will be stored in this folder. The data link will be found in the description below. After loading the data, we need to create a Seurat object. In the Seurat object, we're going to define the project name. You can rename the project into any name you prefer. In this step, we're going to do some basic filtering. For example, the minimal number of cells that express one specific gene needs to be 3. Minimal number of features for each cell has to be 200. The next step, we calculate the percentage of mitochondrial genes. We can do a violin plot showing the number of feature RNAs, number of count RNAs, percentage of mitochondria. You'll see a plot what shows up on the right side shows the 3 features. We can also plot out the relationship between number of RNAs, number of percentage of mitochondria. We can plot out the number of count RNAs and number of feature RNAs together. We can display these two plots at the same time. The results will be shown side by side. The first one will be the percentage of mitochondria versus number of count RNA. The second will be number of feature RNA versus number of count RNA. We can further only keeping the cells satisfy the 3 rules we define here, which is the number of features RNA is larger than 200, number of feature RNA is less than 2500, and percentage of mitochondria less than 5%. We'll do the normalization. The normalization will be a log normalization. After the normalization, we'll need to find the variable features. This is a very critical step for dimensional reduction, and we need to find the genes that preserve more information across different cells. And with this one will return the first 2000 variable features. And we can also shows up the top 10 genes. The top 10 variable feature genes will be stored in the top 10, and using this plot function, we can plot it out the variable features, the mean value, and the variance. Let's take a look. And we can also label the top 10 variable genes. And let's show this plot. On the right side, you'll be able to see the first one and second one. The first one is the variable feature plot. The x-axis is average expression for each gene. The y-axis is the standard deviation or standard variance of that gene. Red color labels the variable genes. Normally we call it top 2000 variable genes. And on the right side, it labels the first 10 variable genes superimposed on this plot. We can also name all the row names, which is going to be the gene names, and store the gene names in the all genes variable. Next, we're going to do the scale data, and the scale features will be applied to all genes. Then we'll do run PCA, which is a principal component analysis, and the features we'll be using are the top 2000 feature genes, which has been found using find variable genes. This one will do a dimensional reduction. You can print out the PCA first 5 dimensions. The first 5 dimensions will be principal 1. This will be the positive value. Principal 2, this is be the component of that specific. After running the PCA principal component analysis, we can visualize the dimension using the visualize dimension loading, and we're going to visualize the first and second dimension. On the right side, you'll see the first component and the second component, and the contribution of the specific genes that contribute to the first and second component. We can plot a dimensional plot of the PCA. You'll see the first component and second component, each dot represent one single cell, and this is how they look like in the PCA plot. You can also plot it out dimensional heat plot. You can see the heat plot for component 1, and you can also see the heat map plot for the first to the 15 dimensions. This is from the first component to the 15th component, and the gene expression in this specific corresponding component. Then we can use elbow plot, shows the component from 1 to 20 and their relative standard deviation. We can see the first component contribute a lot, second still contribute a lot. The higher the standard deviation for the corresponding component means the more contribution they're going to make. Then let's use find the neighbors. The find neighbors will tell us how cells will be similar with each other. The find clusters will cluster the similar cells together. Then we can show the result as UMAP. After running the UMAP, we can show the UMAP plot, which will be colored clusters, and the UMAP will show the results in a UMAP projection. The next step would be define the cell types based on marker genes. Here the clusters were labeled in color and in numeric value. We can use this function called find markers to find the marker genes for the second cluster, or we're comparing the first cluster and the second cluster, or we can use the function called find all markers that will do the pairwise comparison. After identifying all the marker genes, we can assign the top 10 marker genes for each cluster, and then we can do the heat plot showing the top 10 genes for each cluster. This one shows you the heat map plot for cluster 0 all the way up to 8. On the left side will be the gene listed, which is the top 10 marker genes for the specific cluster. You can see for cluster 0, this list of genes have higher expression. Cluster 1 have these genes as a higher expression. Based on the genetic expression profile and based on literature or previous understanding, we can annotate the cells. For example, the first 0 cluster will be labeled as naive CD4 T cells. The second cluster, which is the cluster 1, will be labeled as CD14 positive monocells. So we can use this one to define new cluster ID, and we can use these two commands to redefine the cluster names. Now let's do the dimensional plot again. As you'll be able to see, now the new cluster name has been labeled to the UMAP plot. This cluster is CD14 positive monocyte. You can use ggplot2 to have further control of the plot. First, let's import the ggplot2 and use the plot function, and we can show the plot. In this plot, you can have more control of the plot and by showing different labels of different clusters, and we can change the name as well or change the site. Alright, this is the 10-minute speed run. There's a lot of details in the processing, and I uploaded the detailed explanation for each step in the playlist.